Hello everyone, my name is Findley, and today I will be discussing the role of epigenetics on drug addiction. Today's learning objectives are to understand what epigenetics is and how it works, to understand how epigenetic alterations are produced by drug use, to understand how epigenetic alterations are passed down through generations, and finally to understand how epigenetics can influence anti-addiction pharmacotherapies. Firstly, let's discuss what epigenetics is. Epigenetics are changes in gene expression that are independent of how a person's DNA is sequenced. It is caused by modifications deposited on chromatin in the nucleus of a cell. Chromatin is made of DNA, RNA, and proteins. It is what forms nucleosomes by tightly wrapping around histone protein octamers. One important thing to keep in mind when studying epigenetics is that histone subunits that effectively contribute to the modification in gene expression can be modified. They are modified through several processes that include acetylation, methylation, phosphorylation, ADP ribosylation, and many others, and they occur on more than 50 unique sites. So I'm sure you're wondering, how does epigenetics have anything to do with drug addiction? Well, there are a few ways that we will discuss throughout this presentation. The first being that drug use creates alterations in gene expression. Secondly, when drugs are used in adolescence, the gene expressions produced can be passed down through generation. Lastly, the fact that drugs alter gene expressions mean that the same process can be used to create anti-addiction pharmacotherapies. Now let's talk about how drugs create these alterations in the brain. Drugs that are abused activate the mesolimbic dopamine circuitry. As we've learned, this pathway is from the ventral segmental area, has innervations in the medium spiny neurons, and ends in the ventral striatum, including the nucleus accumbens. Drugs such as opiates, psychostimulants, alcohol, and nicotine increase the total acetylation levels within H3 and H4 histones in the nucleus accumbens. This increase in acetylation levels results in drug-induced alterations to the balance between histone acetyltransferase and histone deacetylase. This manipulation controls the behavior responses of drug abuse. So, moving on. How does drug use in adolescence create transgenerational effects? As we know, the human brain is developing well into your 20s, and that drugs impact brain development, primarily in the regions that affect cognition, emotion, and reward pathways. This in turn makes a person more vulnerable to addiction. It is believed that because the gene expression is being changed during puberty, it affects the gene expression produced on reproductive cells such as sperm, and is therefore passed down to the next generation. It is similar to the study of maternal licking of rat pups discussed in previous lectures. These epigenetic alterations are inherited because of the lack of environmental changes on the germ cell. So, how could the knowledge of epigenetics and how they affect our gene expression be used to create anti-addiction pharmacotherapies? To start, CRISPR and Cas9 techniques can be used to target several genes within the same cell and manipulate the drug-related gene networks. This allows for the isolation of certain epigenetic alterations that cause damaging effects to drug addiction by restoring the normal functioning of certain proteins. This could help diminish things such as binging and relapsing effects of drug addiction. The ability to remove epigenetic alterations can also be used in reverse and add positive epigenetic alterations, such as a distaste for drugs or possibly making you feel nauseated or vomited, along with possible vaccines, as we've discussed previously, to drugs. Well, thank you so much for listening to my presentation. This is the end. Here are my references. I used 2 studies that I found online about these